Hi everyone, welcome to Likey's Academy. This is a second part of DNA replication. Today we are dealing with initial stage of prokaryotic replication. Replication process in eukaryotic and prokaryotic organisms share a lot similarity with few differences. Here we discuss about DNA replication in prokaryotes. Replication in prokaryotes as well as in eukaryotes are divided into three stages: initiation, elongation, and termination. The major things to remember about prokaryotic DNA replications are: DNA replication is semi-conservative, that is, each strand is used as template for the synthesis of new complementary strand. Also, it is bidirectional, that is, the Y-shaped formation of replication fork running in opposite directions. And it is semi-discontinuous, that is, the leading strand copies continuously and the lagging strand copies in segments, that is, Okazaki fragments. In the picture, it's clear that a parental cell DNA replicates with the help of polymerase enzyme and forms a replication fork, and at the end of replication we get two identical copies of daughter cell DNA. The first stage of DNA replication is initiation. As we all know, during initiation the different proteins bind and form complex in the DNA which gradually unwinds the DNA. This is the major thing that is going to happen in initiation. The two components required for initiation are replicator and initiator. Replicator, which is represented as orange color in the image, and it is the entire set of DNA sequence that can direct initiation of replication. It is a cis-acting element, which is they exert their influence only to a region that is physically linked to it. Also, within the replicator, there is a specific site where DNA is first melted and replication begins. This region is called origin or ori. Coming to initiator, it is a protein that recognizes the replicator and recruits other protein complexes required for initiation. They are transacting, which means they exert their influence only to the region that are physically linked to it, but also to the other regions of the DNA. The major two stages in initiation are replicator selection or origin licensing, and second one is origin firing. Replicator selection or origin licensing occurs when initiator protein binds the replicator, and origin firing occurs by the activation of origin by melting the DNA and by recruiting the helicases and other components required for initiation. The entire set of DNA sequence that can be replicated from a replicator is called replicome. Prokaryotes contain a single replicator and thus they have a single replicome. Also, replicator and origin are more or less same region in prokaryotes, and also origin licensing is coupled to origin firing in prokaryotes. The initiator protein in prokaryotes are called as DNA, which has four functional domains. Domain 1 is required for oligomerization of the DNA protein. Also, they recruit other proteins such as DNA-B, that is helicase, and DA protein, that is DNA initiator association protein. Domain 2 is basically a linker domain, and domain 3 they contain locomotive where ATP can bind and have activity of ATPase. Domain 4 that has the ability to interact with replicator. So this DNA protein have two conformations. With bound ATP they are active, and with bound ADP they are inactive. Replicator. In E. coli, the origin is called as ORIC. Replicator is the 245 base pair longer sequence that can be grouped into two components. 9-mer and 13-mer. 9-mer consisting of a repeat of 9 base pairs, and it is the binding site for DNA protein. 13-mer is the repeat of 13 base pairs. It contains 80-rich sequence where DNA first melt. That is called DNA unwinding element, that is DUE. 9-mer, it consists of 3 boxes: R box, I box and Tau box. R box, it consists of 5 motif R1 to R5, where R1 and R4 has highest affinity to DNA. That is, they are very high affinity state. So they can both bind DNA ATP bound condition and DNA ADP bound condition. So R2, R3, R5 are low affinity site. They only bind DNA ATP bound state. Second one is I box that contain 3 motif I1, I2, I3, and they are similar to R2, R3, R5, that is they are low affinity. They only bind to DNA ATP bound condition. Third one is Tau box, that is 2 motif Tau1 and Tau2. They are low affinity, only bind with DNA ATP conformation. During origin firing, DNA ATP protein will oligomerize on 9-mer with the help of DA protein, that is DNA initiator association protein. DNA protein exhibit positive cooperativity in binding. Upon binding DNA, a conformational change in DNA ATP will convert them into a more compact structure which deform and bend the DNA at 9-mer. Bending is facilitated by the other accessory protein like integration host factor IHF and factor for inversion stimulation FIS. IHF protein stimulates DNA bending, whereas FIS protein inhibit DNA bending. The torsional strain created melt 80-rich 13-mer, where other proteins required for initiation are recruited. DNA-B, that is helicase, DNA-G, that is primase, and SSB were recruited. DNA ATP undergoes self-inactivation after initiation. ATPase active site in domain 3 contain the catalytic motif called SRC motif, that is serine arginine cysteine, also called as arginine finger. They are not proximal to ATP in the absence of DNA binding. Upon replicator binding, a conformational change facilitates coordination of arginine finger with ATP and water molecule, increasing the rate of ATP hydrolysis. Thus, DNA ADP will dissociate from low affinity 9-mers. DNA-B, that is helicase. The major function of helicase is to unwind DNA using energy from ATP hydrolysis. This is homohexameric protein in prokaryotes, whereas it is heterohexameric in eukaryotes. They form a ring-like structure with central solvent cavity, and helicases are recruited to single-stranded DNA, and they follow a strand exclusion model for unwinding. A beta hairpin structure in each subunit of helicase binds single-stranded DNA and can adopt 3 conformations based on ATP binding and hydrolysis. ATP bound state they are in top conformation, whereas ADP bound state they are in middle conformation, and in empty state they are in bottom conformation. All the subunit in DNA-B will bind to the single-strand DNA before looping them through the central cavity. The cyclic conformational change in the beta hairpin in all the subunits will severely twist the DNA, which melt hydrogen bonds in front. It results in positive supercoiling in front of the helicase, and it is relaxed by type 1 topoisomerase, that is topoisomerase 1. Helicase move in 5' to 3' direction on template of lagging strand opposite to primase. DNSE, which is helicase loader, it is a hexameric protein and it is also a AAA plus protein, that is ATPase associated with various cellular activities protein. General function of AAA plus protein is to use ATP hydrolysis for the assembly and disassembly of the large protein complexes. So, helicase loader opens the helicase ring and load them into single stranded DNA, and helicase loading occurs only during initiation. Here in the picture it's clear that DNA-B, that is helicase, it is closed or inactive without DNA-C loading factor, and upon binding with DNA-C it opens or it gets activated and it is loaded on to the origin of replication site, that is single stranded DNA. SSP protein, they are single strand maintenance protein and they binds to single strand DNA cooperatively, and they have major functions such as they prevents random nuclease digestion on unbound DNA. Also they prevent renaturation of unbound DNA. Also they prevent formation of intrastrand duplets, and also they interact with other proteins like primase and facilitates their recruitment. DNAG, which is a primase, it synthesizes the primer needed for the replication. DNA polymerase, which is the major replicating enzyme that require a free 3' OH for extension as they cannot initiate polymerization. This short oligonucleotide, complementary to template DNA that provide free 3' OH for extension, is called primer. In replication, RNA primer is used even though it is not an absolute requirement. The two major reasons are: first, concentration of ribonucleotides are higher than deoxyribonucleotides in a cell, and also it improves fidelity of replication. The mass action ratio of ribonucleotides favor high rate of polymerization, and also this greatly increases the rate of initiation event. And how it improves fidelity of replication? You know that different proteins are at interplay during initiation than other stages. So the chances of errors is higher. Because of RNA primer will be eventually removed, any error created during initiation will be automatically corrected. It improves the fidelity or accuracy of replication. DNAG, which is a DNA-dependent RNA polymerase, has three functional domains: domain 1, domain 2, and domain 3. Domain 1, which is a zinc binding domain, is ZBD, which interact with the DNA, and domain 2 have a catalytic site where RNA synthesis occur, and they have metal cofactors present such as Zn2+, and domain 3 contain helicase interacting domain, that is HIV, which interact with DNAB. So, DNAG is a processive enzyme. They have the capacity to add multiple nucleotides upon a single binding to the substrate. DNAG's processivity is very low. About 10 to 40 nucleotides can be added, that is their usual processivity. DNAG creates a short RNA DNA hybrid that is called primer template junction, that is PT junction. It is the PT junction that is recognized by proteins involved in elongation. So, what is a primosome? The multi-protein complex required to unwind DNA and synthesize PT junction is called as primosome. Primosome contain DNAB, DNAG, and SSB. DNAB is helicase and DNAG is primase and SSB is single-strand maintenance protein. That's all about initiation stage. We will be dealing with elongation, termination, and regulation of prokaryotic replication in the next video. If you like this video, please like, share, and subscribe Lycase Academy, your educational catalyst. Thank you.